The overall goal of the following experiment is to identify specific Salmonella antigen alleles. This is achieved by targeting PCR to identify specific sequence differences associated with these alleles. Sample template is added to a PCR reaction mix containing allele-specific primers, and the O, H1 and H2 Allelotyping PCRs are run in the thermocycler. Next, PCR samples are separated by size using gel electrophoresis to determine the size of the amplicon, if present, for each sample or isolate. Amplicon size is then correlated with the expected size for each of the primer sets used in the PCR. Results are obtained that show the identification of specific Salmonella antigen alleles by PCR. Serovar designation is then given based on results for O, H1 and H2 multiplex PCR assays.
The main advantage of the protocol described in this video over serotyping is that it's quick, easier to interpret, and less expensive. This method can help answer some key questions in the field of epidemiology, such as determining the distribution and persistence of Salmonella serovars within or between poultry or dairy farms. The implication of this technique extends towards the rapid diagnosis of salmonellosis attributed to Salmonella serovars, Enteritidis, or Typhimurium. Visual demonstration of this method is critical to understanding how to use this PCR-based method to type Salmonella to the serovar level. Demonstrating this procedure will be Adriana Pedroso and Ying Cheng, research scientists in the Maurer laboratories at the University of Georgia.